The area of research I decided to focus on is that of human trials in brain-machine interfaces. Brain-machine interfaces purport to allow individuals to voluntarily control prosthetic appendages with their minds, which would be very useful for those who have either lost limbs or have become paralyzed. There's been a lot of progress over the last decade or so in measuring voluntary neuronal activity using implanted probes or electrodes into the motor cortex and associating certain patterns in that activity with voluntary movements. This has allowed patients to generally or grossly articulate prosthetic arms and to control a computer cursor on a screen. I'm going to highlight two studies that have been done that made some great progress in these areas. The first study included two tetraplegic subjects, and each had a 96-channel microelectrode array implanted into the hand area of their primary motor cortex. They were shown videos of the robotic arm performing certain motions and were asked to imagine that they were controlling the motions. The electrical activity in their brain was then monitored and recorded by the electrode, and the patterns in the neuronal activity were associated with the motion that they were observing at the time and programmed into a computer. The subjects' implants were then connected to the robotic arms via the programmed computers, and the subjects were asked to imagine those same motions again. And when the computer detected those motions, it would cause the robotic arm to move in the same way that the subjects observed when they were first imagining those motions. So by the endpoint of the trials, both of the subjects had experienced success in reaching and grasping with the arm, and one subject experienced a success rate of 75% and the other 95%. So the image to the right is one of the subjects being able to control the robotic arm to reach for a bottle of water, to grasp it, pull it towards her, and then take a drink, and then reach and put it back, which is, obviously a great success. In the second study, which was done by some of the same researchers, but several years later, they included 3 subjects who were suffering from paralysis, and each one also had a microelectrode array implanted into the hand area of their primary motor cortex. This time, though, they were connected to an amplifier and then to a computer, and it was programmed to move the computer cursor in one dimension on the vertical axis in response to certain patterns of nerve impulses. And instead of the subjects being shown movements of the cursor on the screen and then thinking that they were controlling those motions, the subjects were instructed to just imagine themselves performing a random assortment of voluntary motions while also staring at the computer cursor, which was responding to those motions. And they were asked to observe and correlate the motions that they were imagining with whatever motion the cursor did in response. And so they were able to learn to move the cursor by imagining certain specific motions. At the end of the study, all 3 subjects succeeded in controlling the cursor in one dimension. 2 of the 3 subjects achieved a 100% success rate, and the other subject was able to control the cursor in 5 of the 9 sessions that they did. And so the diagram here is the track. The blue line is the track of the computer cursor, and the green box is where on the screen a target was placed that the subjects were instructed to direct the cursor toward. And so you can see that as the target moved, the subjects were able to direct the cursor to follow the target. Going forward, there are some challenges. There are many challenges that still exist that need to be overcome. And some of them are as followed. Specifically, getting more fine-tuned motor movements, such as precise movements of individual fingers, have been really difficult. And some causes that are proposed are that there are just simply structural differences between a natural human hand and the robotic prosthetic, which might be hindering the brain's ability to learn to move those finer components of the robotic arm, at least over the lifespan of the studies. There's also a limited resolution of the microarrays, which could be limiting how much precise information the computer can measure. There's also a limited lifespan of the implants themselves. Some of the studies lasted for years, but at some point the implants will either fail or need to be removed for another reason. And so that limits the amount of time that subjects can practice and learn. And also they're only in the laboratory for a limited amount of time in each session. So perhaps with more time, they will be able to achieve a better definition of movement. And then they also have a lack of feedback. So the robotic arms or the computer cursors don't provide proprioception or other sensory feedback to the brain. So the subjects can only learn the effect of their voluntary thoughts on the motion of either the cursor or the arm by observing it visually. And so that lack of feedback might be hindering their ability to learn to control it more precisely. So these are all areas for future research. If these can be improved on or refined, then we might be able to allow patients suffering from paralysis to have more precise control.